Why is cell polarity so critical for hematopoietic stem cells function upon aging and rejuvenation? So hematopoietic stem cells, like other somatic stem cells, need to critically balance the symmetry of division, in that they need to be able to self-renew, but also to differentiate. And this is what is so-called an asymmetric division. Intuitively, polar cell might be more prone or able to divide asymmetrically. We investigate whether this was true by comparing in pair-daughter single-cell assay the ability of young and aged HSCs to divide asymmetrically. We did perform this assay by sorting young and aged single-cell, monitor their division in vitro, and then investigating how they distribute molecules during division by 3D immunofluorescence analysis, and also by separating daughter cells immediately after division and analyzing each daughter pair by single-cell RNA-seq, single-cell ATAC-seq profiling, and also by single-cell transplantation. The results clearly show that young hematopoietic stem cells divide more frequently asymmetrically, and this was based on the amount of histone 4 K16 acetylation inherited by the two daughters during the division. And mainly, young daughter pair were already asymmetric, with one daughter having more histone 4 K16 acetylation than the other during mitosis, while aged hematopoietic stem cells divide mainly symmetrically. This data on this epigenetic asymmetry during division is based on histone 4 K16 acetylation amount were also confirmed by single-cell ATAC-seq profiling of the two daughter pair. And again, we detect that aged stem cells divide more symmetrically as based on single-cell ATAC-seq profiling, in that the amount of open chromatin region detected in aged daughter pair was more similar between the pair, while in the case of young, the situation was that most frequently one daughter cell was having much more open chromatin region than the other. We also define qualitative differences and similarities between young and aged daughter, and major similarities were about metabolic pathway, where glycolytic pathway were enriched in both young and aged stem cells, while major differences were in pathway linked to signaling, mainly showing that there might be indeed a significant difference in the ability or in the predisposition of young and aged daughters to interact with the microenvironment. Finally, single pair daughter cell transplant functionally confirm that young hematopoietic stem cells divide mainly asymmetrically, while aged one divide symmetrically, again supporting that the epigenetic asymmetry is indeed parallel by a difference in the function of the single daughter pair.